Hello and welcome to this design practice 2 module 12. We will be continuing on the topic of micro electromechanical systems or MEMS. So, we were discussing about biochips or lab on chip devices, and basically if you may recall, lab on chip is a term for devices that would integrate multiple laboratory functions on a single chip with extremely small minuscule fluid volumes. There are applications in the area of research related to medicines. For example, pharmaceutical research deploys quite a bit of this lab on chip for studying issues related to protein chemistry or protein fouling or associated technology models. A lot of these chips are often on used in food safety, particularly food and water safety, which are considered to be the borders, which would be agents of carrying infection into the human systems and somehow need to be protected. The advantages that lab on chip have to offer are many. For example, they are away from human intervention, fully integrated in nature, highly automated, highly sensitive, very rapid to work in, and then you talk about the cost performance product of this chip. So, they are low cost and high performance. So, they are also one of the object of central theme or attention of a lot of companies. So, some companies which sell these technologies often are, for example, Nanogen, Caliper, Affymetrix, Aclara, so on and so forth. And by and large, a major portion of the lab on chips are still dedicated to the area of integrated gene analysis, although there are technologies which have evolved which are considered to be immunochips, where ELISA and some other assays are carried out. Okay. When we talk about a step further and hit upon the domain of independent cells and their behavior. So, we are all aware that the human cell, or let us say any mammalian cell, is a fantastic piece of machinery, because it does compress information which is stored somewhere in the chromatin region in form of chromosomes or DNA into sequences which can then mRNA sequences which can then lead to the production of proteins at they produce proteins in the otherwise well known protein warehouses which are present in the perinuclear region of a cell, and these mRNA molecules typically take the information which is generated by a signal which is carried into the nuclear region by a sigma factor, okay, which typically associates with the RNA polymerase, an otherwise very well known enzyme, and tries to compress information from the DNA to the RNA. So, it is a very complex process of transcriptizing the sequence information which is there on the DNA of a cell. Probably some of you are also aware of the fact that once the mRNA is made and then the mRNA goes into the perinuclear region in those protein warehouses which are floating around, otherwise known as endoplasmic reticulum, there is a change of the language from the normal sequences on RNA to sequence of amino acids which are otherwise well known as proteins, and these proteins are what determines the functionality and behavior, the physiological aspects related to a living cell. So, proteins control quite a bit of transcription of specific genes, and such movements of DNA to RNA and then to proteins are being controlled, okay, in real time through in a very close spaced way, because whatever expression changes happen on the surface of a cell can be monitored on a real time basis through, again, using MEMS platforms. Okay. And so, some of these tools may be an array of MOSFET devices which are going to measure the change in current on the basis of surface potential changes across the surface of a cell as it expresses proteins, etc. They are all in place to tell about this roadmap and analyze how single cells would behave and understand the various signaling pathways within the cells which would lead to such a transcription and translation mechanism. Okay. So, it is of immense utility to cell physiology, particularly to understand how they behave with stimuli under different conditions. And so, this whole area of nanobiology in a way uses such microstructures created and printed in a very array like manner to understand more about how much expression is happening on surface of the cells, etc. So, this are one of the novel tools for nanotechnology. Also better known as nanobiology. There are dedicated centers to studying of how cells would change expressions, etc., based on either electronic responses or fluorescence responses, and it is worthwhile to use MEMS in these kind of sensing mechanisms. So, today talking on the size and scale, obviously when we talk about merging these two worlds together, there are different biological moieties, for example, on the left side of the scale here, varying from even the atomic level of tens of angstroms all the way to 1 angstrom, all the way to about plant and animal cells, which are quite high in size, maybe 20 micron plus. And on the left, the world that relates to how microstructure can result in some of the features and structures of a size parity with some of these biological features and structures. This, for example, is a double helical structure of a DNA. This is a virus molecule, a few 100 nanometers in size. This right here is an SEM image of an E. coli bacteria. And so, therefore, when we talk about such a merger between the fabricated structures and the naturally available structures, one thing which is very clear is because of the very minuscule contribution of its own mass or weight related to such structures, it is always a very nice merger, and it is always a very sensitive detection. Okay. So, therefore, technologies have evolved on 2D CMOS platforms, which have all these different devices integrated, including microfluidic MEMS, molecular specific sensors, MEMS and NEMS, which are of the structural nature, and then of course, molecular devices and memory together to give you some idea of how these entities do behave in different environments and what are the kind of stimuli that they are able to generate. So, these typically form the area of what you know as integrated biochips. Okay. So, both the macro, micro and nano area, and there is a huge amount of demand of such technologies. They are rapidly emerging around the world. Part of it also is reflected under the overall broad stream of biodesign, and so therefore, it is of quite a bit of significance to a product design engineer as well as a biodesign engineer, okay, who can take these technologies further for very simple, easy to use products which would serve the diagnostic needs for the human society. So, when we look in a nutshell this whole area of micro and nano systems and how it has evolved, a lot of to and fro inspirational learning has happened in both areas. For example, there has been devices which are made through biological entities. Look at DNA based nanowires or molecular nanowires, which are inspired for some of the next generation molecular electronic devices. They are highly inspired from what happens in biology and biomedicine, and it gives something which is of significance to the area of micro and nano systems. Then we talk about self assemblies, both DNA and protein mediated or self assembled monolayers, or if we talk about inspirational learning borrowed from the skin. Look at the skin, for example. It is such a wonderful thing. It can sense, it can respond, it can heal, and so if you look at the skin from a perspective of an engineering object, it is a dense array of sensors integrated together into a small area, and so therefore, it can of course inspire a lot of learning experiences for micro nanotechnology, where you can have bio inspired materials or skin like materials, okay, which can serve the humanity better in general. On the left side here are some of the tools that we have taken up from the micro nanosystems technology, and it helps in really the bio inspired world. And for example, we talk about hybrid bio devices where structuring at the micro nano scale are used heavily for doing tissue engineering applications. There are scaffolds which are grown, which are of sacrificial material, and they are biocompatible, and there is cell growth promoted within such scaffolds, and then later on the scaffolds are etched out or pulled out, so that you have a hollow structure in between. Similarly, there are the therapeutics associated with drug release, targeted delivery. Today we can actually make a carbon nanotube with functionalization both internally and externally, and you can flow this through intravenous means to a certain site which is affected with some tumorous growth or maybe cancerous growth, something like this, and then that way drug delivery can targetedly happen for therapeutic purposes. Again borrowed inspiration from the micro nanotechnology. You can also borrow a lot of inspiration from biochips and what quantum dots or silicon nanowires can do in terms of recognition, which can directly help the bio world. So, there is a lot of learning from micro nanotechnology which can even translate into biology and biomedicine. So, on the right side here, which is learning borrowed from biology into micro nanotechnology, would produce some novel solutions and some for some frontiers in materials and information processing systems, whereas when the learning is reversed, that means you are learning from the micro nanotechnology systems and applying to biology and biomedicine, there can be some novel solutions and frontiers in medicine and biology. So, that is how we can actually look at it as a synergistic system, and all these are based on or rotating around that fundamental micro and nano systems. So, there are some other examples I would like to look at very closely, which are found abundantly in the areas of microsystems for biology. For example, look at this neuroprobe which has been developed by Dr. Weiss's group at the University of Michigan. It is about, it is a probe which would have a lot of printed electronics and would be able to monitor the electrical activity of the brain tissue, particularly in rat brain. And for using this probe, the incision that is needed to insert this probe into the brain is very small, because the overall dimensions of this probe is also quite small. It is again borrowed as an inspiration from the area of micro nano systems, but it can do something like painless monitoring of the electrical activity of brain tissue, for example. This again is another very nice example. This is a neuroprobe developed at Stanford, which typically was supposed to do deep electrical stimulation, which is a again a therapeutic strategy given to particularly Parkinson disease patients, and the idea is that if you can make this experience painless for them, it is going to be of immense value to the society. These are a set of neuron cells growing on bunch of MOS transistors, which are micro patterned on a surface, and this is the area of nanobiology I have described in the last step, where whatever happens on the surface of the cells and the understanding that you develop here is psychological or physiologically how the cells would respond in proximity of each other or while they are spread apart on a surface, and how they cross talk with each other. So, this is again another beautiful example of how micro nanotechnology can be inspired for something directly translating to the biology or biomedicine. This right here is a micro needle. It is again another example which can be how micro and nano systems go into helping the humanity through interaction with the human side. We all know how painful it is to inject something intravenously into babies, for example, or even into old age people or normal human beings. Some of the people are very afraid of painful injections. So, this injection right here is rhythming very well with the with the injector on a mosquito, and therefore, just as a mosquito bite, when the mosquito bites you, you do not feel the pain, because the needle which goes, the mosquito needle which goes into your body typically goes in a region which is just above the vasculature, where there are pain receptors, and is not able to damage, deform those pain receptors, which otherwise would be the case when you try a bigger needle. So, this right here is another example of a silicon biochip which has been developed at Purdue for detection of food microorganisms. There is another example right here about a series of micro cantilevers which have been developed by IBM Zurich research, which talks about how cantilevers and bending in cantilevers can be mapped to physical reactions, chemical reactions which happen on the surface of these cantilevers. So, these are various examples of how microsystems can be manifested within the field of biology and biomedicine. A small take on further applications is this domain of microfluidics, which have come up because of the advent of the biochips and the diagnostic assays. So, if we look at the area of microfluidics, it is the transport of fluids at the microscopic length scale, And there are many properties of such flows. For example, there are surface effects which become prominent with high surface area to volume, low thermal mass and high heat transfer capability within these channels, low value of Reynolds number. Okay, so these are some of the associated aspects of this field of microfluidics, where laminar flows, because of the low Reynolds number values, lead to mixing which is only diffusional in nature. There are hardly any eddies or vortices which are generated within the flows. This simulation right here is 5 dyes going end to end parallelly in a microchannel. So, the Reynolds number, which is actually the ratio between the inertial and viscous forces, is usually dependent on the average velocity, the length scale factor, and the viscosity of the medium. RE is usually less than 100, very often it is less than 0.1 when we talk about microfluidic devices. And so, these colors here, as you are seeing, although they are flowing past each other, they can be extracted as is without much mixing, because most of them are having almost zero concentration difference. So, such experiments are even possible at the microscopic length scale, where we can talk about such microfluidic devices with peculiar properties which can be used for many applications. So, we will now try to look at a slightly different aspect, which is about how to design sensors and what really are sensors. And so, definition wise, if we look at sensors, it is really a device that detects or measures a physical, any chemical or a biological property or entity, and records, indicates or responds to it. Sensors can be classified into all different physical, chemical and biological sensors depending on the property that is sensed. Physical sensors, for example, sense physical properties like temperature, distance, mass, pressure, etc. Chemical sensors measure chemical substances by chemical or physical responses. Biosensors, of course, measure chemical substances by using a biological sensing elements. And so, it is all about what exactly is that element and how there is a change in the signal, how there is a transduction for the sensor to start detecting various responses. So, when we talk about a schematic of how a sensor should look like, and this is more prominent towards when we talk about chemical and biosensors particularly. So, there are very many components of such a sensor. For example, let us say there is an analyte of interest which you want to detect for a certain specific chemical, and let us say the sensor which is built here has a chem biodetection element which is specific to a certain chemical, which it would bind to the analyte and thereby produce a response which converts or transduces into something which is machine readable. Now, chemical signal converting into an electrical or an optical signal is a very commonplace affair which happens in almost all of molecular identifications which are carried out. So, such a change in response, for example, let us say we are talking about a gas sensor. So, if there is an element which takes up a certain gas from a set of gases within the environment and changes some property, okay, the property could be let us say it starts releasing electrons, thereby making positive ions and changing the conductivity because of such release of electrons into the environment because of the reducing gas that comes in. Okay. So, it can detect a reducing gas by change of resistivity. So, this transduction that is happening at the interface is because of a phenomena of reduction which is happening because of a electron getting donated, and change in conductivity owing to this donation of the electron in the presence of a certain gas. So, the gas creates a physical change which creates a signal further, an electrical signal in this particular case, which can tell you about how much gas was present. Okay. So, they are quantifying the signal, the electrical signal in terms of the concentration of the gas that was present. So, this is the whole modality or scheme of how sensing can be carried out. So, you are transducing from chemical to let us say electrical in that particular case, and this signal is now recorded somewhere. It could be a computer which records the signal, or maybe it could be some kind of a storage device which records the signal, and it gives you an idea about what is the concentration of the analyte gas which was there in the system because of which this particular signal was generated. There are many examples of this like which are continuously seen. For example, the simplest case could be that of a litmus paper or a pH indicator electrode. So, here what is happening is there is an analyte which is a solution of which the pH is to be measured. There is a detection element, it is a chemical dye on the litmus or a set of chemicals in the pH electrodes. There is a transduction which is a change of color now, because the pH paper changes in red color, becomes red color or blue color. So, change of color because of the change in absorbance of the visible light by the chemical or change in voltage generated by an electrode. Either way, this is caused by the presence of hydrogen ions, okay, which can detect whether how acidic or how basic a solution is. And then the signal processor, for example, in the case of a color change, is your brain, which records the color change. You are able to see the color change through your eyes, okay. And if I looked at how my eye functions, it is also a sensor, or how my nose functions, that is also a sensor. I am going to come to that level probably in the next lecture, not in this lecture, but this is how you record the simple litmus paper, the dye on the litmus paper being the recognition element, and the dye changing color, which is again made visible to the eyes, and eye being the recording device, makes the whole sensory system sense the hydrogen ion concentration. So, there are numerous such examples in how a chemical and biosensor can be defined, but what I am going to do is probably in the next lecture I am going to start giving some description about how our basic senses, like the sense of smell or the lens, let us say the sense of visualization, is basically outlaid as a sensorial model. So, I will close this particular module here and meet you in the next module. Thank you very much.